The second complexity is that the primary transcript, that is hnRNA, heteronuclear rRNA, contain both the exons and the introns, and these are non-functional. So there is like processing, that is splicing. Now where the introns, that is intervening, that is unnecessary sequence are removed, and exons which are coding, which are necessary, are joined in a defined order. So HNR undergoes additional processing for this, called as capping or tailing. So capping, unusual nucleotide methylphonosine triphosphate, unusual nucleotide, right, is added to the 5' end of the hnRNA, and in tailing adenylated residues, that is 200 to 300, are added at 3' end in a template independent manner, right, in a template independent manner. Okay, so it is fully processed hnRNA, now called as mRNA. Now fully processed RNA is mRNA, that is transferred out of the nucleus for translation. Now the significance of such complexity is now begin to be understood. So the split gene arrangement represent this split gene arrangement represent probably an ancient feature of the genome. The presence of introns is reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. The presence of introns is reminiscent of antiquity, and the process of splicing represents the dominance of RNA world, right? So genetic code. Okay, so come to the point, like George Game, who argued that since there are only 4 bases, if they are to code for like 20 amino acids, the code should be made up of 3 nucleotides, 4 power 3, that is 4 into 4 into 4, and it would generate like 64 codons, generating many more codons than required. Actual amino acids are 20, and 61 are like coding for amino acids, and 3 are stop codons, right? And now Hargovin chimera, the chemical method. Okay, so providing proof that the codon was triplet. Now how they give the proof. Now the first one is the chemical method developed by the Hargobian Khorana was instrumental in synthesizing RNA molecules with defined combinations of bases, homopolymers or copolymers. Now Marshall Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. So Marshall Nirenberg's cell-free system for protein synthesis. Then Sever-Oka enzyme polynucleotide phosphorylase also helpful in polarizing RNA with defined sequences in template independent manner. That is enzymatic synthesis